In the previous video, we used the blast search algorithm to find insulin orthologs in different species. And we made a table like this and showed that the best match, best sequence match in all these different vertebrates, when further studied, is shown to be a gene that codes the insulin protein and does that same job that insulin does in our body in all different vertebrates. Uh, that led to this interpretation that we have an insulin gene that is similar but somewhat different in our bodies compared to a chicken because we share a common ancestor that already had that ancestral insulin gene function, and our modern insulin genes, these derived genes, came from that common ancestral gene, and that's why they have such great sequence similarity, because they used to be long ago identical. And again, we define this important term ortholog, which are two genes in different species that share a common ancestral gene. Now we're going to do a blast search for insulin genes by comparing the human insulin gene with other genes in the human genome. So instead of looking at different species, we're just going to go and look within the human gene using the blast search and the insulin gene as the query. And so what we find now is that the best match within humans of the insulin gene is, of course, the insulin gene itself. And that's shown at the top of this list. Then there are two genes below it, the insulin-like growth factors, gene 2 and 1, that code for proteins that have a lot of sequence similarity with the human insulin gene. So again, this is searching the human genome for matches to the human insulin gene. And we find one perfect match, yeah, that's insulin. And then we have these two other matches that are very similar. We call that a gene family, and we'll talk a lot about gene families in this course. When we study the function of the proteins coded for by these genes, the proteins are going to do different jobs. But we'll see that there are overlaps and relationships. And again, we'll talk about a lot of gene families in this course, and we'll see that they have overlapping yet distinct functions for each of their proteins. Here's just the readout, the details of the matches, and you can see that the top match is basically a perfect match. This is showing the single letter abbreviation for the amino acid sequence coded for by the human insulin gene matched to the human insulin gene. Then down below you can see that there are similarities between insulin and the insulin-like growth factor 1 and 2 proteins. Here's how we think about gene families. We think, well, yeah, there's a human insulin gene, and then the gene family that includes the human insulin gene includes some other genes. In our body, the human insulin gene family has 3 gene members: insulin, insulin-like growth factor 1, and insulin-like growth factor 2. 3 different genes. You got 3 insulin family member genes from your mom and 3 from your dad. Alleles of those 3 genes. Here's what we think about. That this gene family arose because in our ancestor there was a kind of mutation that's called a gene duplication, where when the DNA was getting copied by accident, it copied the human insulin gene twice. So our ancestor had an ancestral version of the insulin gene that got copied twice, and now we had 2 insulin genes. One of the insulin genes retained that ancestral function, and you can see that our modern screwdriver looks basically identical to the ancestral screwdriver. That's because that's an important job. Insulin does an important job, and if that gene's ever mutated, the person has a problem like that. So there's selective pressure to maintain that gene as is and that gene function as is. And there could be some slight differences over time, but there's a lot of pressure to maintain that. On the other hand, the second gene doesn't have that selective pressure, and starting at that point can evolve a new function. And that's a pretty important concept in evolution in general, and certainly developmental biologists will talk. We'll talk a lot about gene duplications leading to gene families. Here we have another term, and that is this. Paralogs are two genes in the same species that share a common ancestral trait. So we've got orthologs, we're thinking about human and chickens. We got paralogs, we're thinking about gene families within one species. Again, when we get a gene duplication, one copy of the gene retains the ancestral function, like insulin, and the other copy of the gene can evolve a new function, like the insulin-like growth factor 1 and 2 genes. Several duplications over a long time can create multiple paralogs, and that's what we call a gene family. Gene families all look similar because they have that same ancestral function. So they look kind of like screwdrivers, but we can get this great diversity of screwdrivers. And we see that a lot in development, where an ancestral species had a sort of a similar or simpler toolkit than some modern species. Through duplications, we've got a variety of different members in a gene family, just like a complex toolkit can have a variety of different screwdrivers. Here's a really interesting concept that will help us a lot in this course, and that is that over time, the gene family members can be quite diverse. So we can have a gene family that includes screwdrivers and shovels, for example. They do really different jobs, but through this graphic you can kind of see how if you had an accidental mutation that led to a gene duplication, you could retain that ancestral function, but then that second gene could evolve just by chance mutations over time into sort of a funny shaped screwdriver, and then a really weird shaped screwdriver, and then a shovel, just by extending the handle of the screwdriver and expanding the blade of the screwdriver.